Good morning, everyone. My name is Chandra from Professor Zhuwei Zhang's lab in the School of Computational Science and Engineering at Georgia Tech, and we'll be presenting our recent work on comparing deconvolution and label transfer methods for spot-based spatial transcriptomics to optimize experimental and computational practice. So first, starting off with spatial transcriptomics. Spatial transcriptomics is just a technique that preserves gene expression with the spatial location of the required spot or cell. The most common type of spatial data is spot-based data, where each spot may contain more than one cell. In this case, we have 7 cells, and this requires further processing for such as deconvolution for downstream tasks. Now we also have single cell data as previously mentioned, but this comes with a high cost, and we would need to consider the trade-off between the resolution and the cost of obtaining our spatial data. So the objective of our study is to compare methods for deconvolution and label transfer, and then to assess spot sizes for the most optimal spot size for each task, and then provide guidelines for each. So coming to an overview of our study, first we have the single-cell spatial data, which is cell type labeled, and then we perform spot simulation on that with different size spots. So here we consider spots ranging from 3 to 200 cells per spot. Then we perform 2 tasks on the spot-based data, which is deconvolution and label transfer. So the first deconvolution is breaking down the spot into its constituent cell types, which we label as task 1, which is to determine the cell type proportions within spots. And then we perform label transfer, which is to assign each spot a single cell type label. Both of these tasks require a reference data, which in this case we use a single cell RNA-seq data, which has cell type labels. And So for the methods considered in this study, we consider the top methods according to the recent benchmarking papers. For the deconvolution methods, we consider the top 3 methods according to Lee et al. 2023, which are card, tangram, and cell to location. For the label transfer methods, we consider the top methods from Abdullah et al. In 2019, which are SVM, Singlar-Surratt, and SCBI. So we considered 4 datasets in our study, the first being Murfish mouse brain, which we will be naming as Murfish 1. The second is a StereoSeq mouse embryo, which we name as a StereoSeq MOSTA. The third being Seqfish Plus of the mouse olfactory bulb, which is a Seqfish Plus. And the last one being a Seqfish mouse gastrulation dataset, which we name as Seqfish gastrulation. So coming to the first step in our study is to simulate the spots. To do this, we take the single-cell spatial data and then overlay a grid on it. And then we consider the maximum inscribed circle for each. Consider the maximum inscribed circle for each grid, and consider that the cells inside those circles are the ones that constitute the cells within those spots. So the cells that are inside those circles, the expression of those cells are aggregated to form the expression of each spot. And this is repeated for different spot sizes, and we get data sets for different spot sizes. So coming to how we evaluated these metrics, we used the evaluation metrics that was used by the benchmarking papers previously mentioned. So for the label transfer task, we considered F1 microscore. And for cosine similarity, for deconvolution, we considered cosine similarity. A limitation that we found of these methods is that they tend to give high accuracy even when the prediction is not very diverse. So, for example, we have here single R on the left and SVM on the right. And we can see that single R prediction is much more biologically relevant, but it gets a lower accuracy compared to the SVM prediction. So we considered this problem and we tried to fix it. And so the solution we came up with was to introduce an evenness score that was based on the Shannon entropy. So this Shannon entropy is calculated for the sum of proportions for all spots. So we have an example here where case 1 and case 2, we have a matrix with spots being the rows and columns being the cell types. And in case 1, we have the each spot belonging to different cell type. And in case 2, we have spot 1 and spot 2 belonging to the same cell type, and spot 3 belonging to a different cell type. So taking a sum for all rows, we have these vectors for which we calculate entropy on. So the entropy for case 1 will be more than the entropy for case 2. So using the entropy measure for our metric, using those two, using the accuracy and the cosine similarity on the entropy, we get the adjusted metric, where the adjusted metric for the single R is greater than the SVM prediction, which is much more reasonable. So finally, coming to the results on the deconvolution task. This is for one dataset, the Seek-Fish gas relation dataset. We can see that the deconvolution metrics here in green and blue all increase with an increase in spot size, which goes linearly with more than 25 cells per spot and increases exponentially from 0 to 25. So it can be seen that a spot size of 25 or more cells per spot would be ideal. And coming to comparison for all methods on all the data sets, we can see that tangram in blue is the best performing method. And the second task we considered was the label transfer task. We have tangram in green performing the best, and we can see that the small spot sizes actually perform the best here. And actually tangram, which is a deconvolution method, outperforms the label transfer method, which is interesting. So a comparison on all the methods, we have tangram in blue performing the best even for the label transfer task, and we have the single R coming in second place. So finally, the recommendations for both tasks. For deconvolution, we recommend a spot size of 25 or more cells per spot, and tangram is the best performing method. And we can see that there is a sharp improvement in accuracy once you increase the spot size from 1 to 25 cells per spot. And for label transfer, you can see that the spots with 10 or less cells are ideal, and tangram and single R perform the best here. So we also wanted to measure how changing the dataset would affect the methods. So we considered two metrics to measure how the data was distributed according to cell types and the position. So the first metric here is spatial domain segregation index, which measures how cells of the same type are distributed in space. So a low value of this metric would indicate that cells of the same type are clustered tightly together. And the mean nearest neighbor is just measuring how close cells are in general. So coming to the correlation plots, we can see the one circled in neon are the statistically significant values. And we see that tangram and single R are not really affected by changes in data sets. But CARD here has a high correlation for spatial domain segregation index, which may suggest that CARD tends to prefer data sets that are pretty spread out according to the cell types. So finally, concluding our presentation, we have given the optimal spot size and the method for each task, which is for deconvolution, we have the tangram as the best method and 25 cells per spot as the recommended spot size. And for label transfer, we have tangram and single R as the best method with less than 10 cells per spot. And we also introduced an adjusted metric to provide balanced evaluation, and certain methods are more sensitive to changes in dataset than the others, as seen in card example. So limitation of this might be that the simulated spot might not capture all the complexities of the real experimental data, but there is nothing we can do about that right now. So finally, I'd like to thank Professor Xue Zhang, our corresponding author, and the PI, and all the members in my group who helped me with this project. Thank you.
Quick question. So for deconvolution, because there's already published like benchmark study suggesting the RCTD, and now it's called SpaceXR, has the highest performance. But in your analysis, did you include RCTD for the benchmarking?
We considered card and tangram because they were the best ones that were given by the deconvolution benchmark. So a further analysis would include RCTD. We haven't considered that yet. Yeah.
And what's the rationale of like repeating this benchmarking study?
Oh, this is for including how the deconvolution method responds to a change in spot size.